All right, Nicole, I think I'm gonna get started. Just so that people don't have to sit here listening to this deafening silence. If that's all right. So thank you all for joining, and those of you who are still being virtually zoomed into the room. I'm Todd Alexander. I'm the Associate Director of WCHRI and a pediatric nephrologist at the Story Children's Hospital, and I welcome you here this afternoon. Thank you for attending our, I forget which number, I think this is our third in our Precision Health Seminar series that has started this year. For those of you who don't see in the chat box, please note that the session is being recorded. I also respectfully acknowledge that the University of Alberta, and where I'm sitting as Dr. Kalaseru, is on Treaty 6 territory, which is a traditional gathering area for a number of First Nations, Inuit and Métis peoples. I'd also very much like to thank, at the same time, our funders, including the Stoley Children's Hospital Foundation, of which some of the board members I think are here, as well as the Lois Hole Hospital for Women, who made this session and a lot of the work that we will be describing, or Dr. Kalaseru will be describing today, possible. So thank you very much. And with that, it is my sincere pleasure to welcome Juana Kalaseru to present on a topic that she's wrestled with, but we've also wrestled with a lot over the course of our, I'm guessing, 8-year relationship now. So Dr. Kalaseru is currently an associate professor of medical genetics at the University of Alberta, and I'm going to call her a staff medical geneticist in the children's health sphere at Alberta Health Services. Although that it's a bit unclear, the reporting lines there. Regardless, the majority of her clinical work focuses on diagnosing and helping children with genetic diseases. Dr. Kalaseru's training path is rather complicated, and I hopefully will articulate it in such a way that makes sense. She did her medical training, her MD in Romania, her native Romania, and then she went to Rome for a number of years where she did medical genetics clinical training, after which she was recruited or went to the University of Toronto in the Hospital for Sick Children, not six, where she had more genetics training and some research, after which she went to do more medical genetics training at the University of Calgary and research training at the University of Calgary, which was funded by a WCHRI Institute of Maternal Health Fellowship. Before in 2012, she was recruited here and promoted to associate professor. She's held founding from Rare Disease Network, CIHR, and local and provincial authorities, and published a number of studies, and I think she's going to describe one of them in her talk today, describing variations in children's DNA that have resulted in genetic disease. And hopefully she'll describe how we can do this better and faster going forward in the new year. And I very much look forward to this description and your ideas, and I valued your collaboration and discussions over the years. So with that, Juana, take it away, please.
Thank you very much, Todd, for your kind introduction, and thank you for this opportunity to present our project. Wouldn't happen just for, by me being in my office thinking about it. So it took a lot of people, even to this point, to be where we are today. And thank you very much, everybody, for joining to hear about this. Many of you are my collaborators over the years, so I'm particularly delighted to not see you, but at least see your name on the screen. So I'm going to try to share my slides. Okay, so can you see them? Yeah, okay, very good. So, for me, I'm a pediatric geneticist and I do prenatal diagnosis. So I have to say that from the beginning, WCHRI has been my natural home because I really migrate in my clinical and research work between child and women's health. So these are my, I can't really move my slides for some reason. Okay, these are my objectives. If you didn't have a chance to see them before, I'm just going to briefly describe what has happened in our field, in especially the last decade. I'm going to give you an example of how a real life project may work in medical genetics, and how that actually has inspired this initiative of the Translational Genomics Hub that WCHRI supports. And I'm very grateful for that. So, precision health is a hot topic, everybody knows nowadays, and I'm very excited about it because for someone like me who has been in the field of medical genetics for about 20 years, the progress that has happened in genetics, while highly desired, has been unprecedented and unexpected. We used to be these kind of physicians, kind of located in an ivory tower, with a bit considered eccentric, who will have all these esoteric thoughts about patients. And because of the technological progress that has happened in our field, we have been pushed to the center stage. And in this context of the precision health, an initiative that has been embraced by Alberta Health Services, by our university, by our research institute. So here on the right-hand side, you see this document that has been produced in Alberta, and that addresses these challenges to implement precision health. And a sentence caught my attention from the beginning. If you haven't read it, I really recommend you do so. Alberta's innovation networks are too fragmented with too much competition and not enough collaboration. So, when it comes to precision health, nothing is more personal to you than the DNA. So I think we fit perfectly in. The DNA, needless to say, is at the core of our existence, but we are very much not aware of it most of the time. I like to compare the DNA with the environment. So it is there all the time. We normally ignore it, and we take it for granted. And it takes really like a catastrophic event that will be, for the environment will be a hurricane. For the DNA will be a mutation that will make us become aware of the consequences of these changes. So if you are sitting in your chair right now and you are hearing me, you are looking at the slides, you are trying to figure out my accent, you're trying to integrate what I'm saying, it is because there are a myriad of proteins derived from your DNA that are interacting with each other and hard at work, all deriving from these incomprehensible complexities of this 2-meter-long molecule made of 4 alternating letters, G, C, A, and T, that form the 46 paired chromosomes that have to fit tightly in the nucleus of each cell. So the DNA has these 3 billion letters, and only about 1.5% represent those coding regions of the 20,000 genes that we have in our DNA. So it is quite an extraordinary event that all of us are actually here, because we could have been easily a miscarriage. This is a very frequent event for humans, and a lot of the causes are genetic. We could have been affected by a very severe mutation in a developmental gene that would have made us be a demise, a stillbirth, or a newborn that was born with one or multiple severe major congenital anomalies with potentially severe long life challenges that would have affected that patient, but also their family, their parents for the entire life. Now, aside from that, we all escaped that possibility, luckily, but all of us carry millions of variants in our DNA that are just normal variation. And these contribute to how tall we are, partly for our weight, of the skin and hair color, how we metabolize our food or medication. So why we are all a little bit different from each other. However, this difference is about 1% of our DNA between any 2 people on this planet. All of us also carry a number of recessive mutations that will not have an effect directly on us, but it would take that one other person who carries a mutation in the same gene like you to increase your chances to have a child with recessive disorder. And all of us, again, are carriers for a number, unknown number at this point, of susceptibility genes for all sorts of complex disorders that sooner or later are going to catch up with us. Helped obviously by our lifestyle choices as well, and help us develop cancer, diabetes, high blood pressure, so on and so forth. So you see that that concept of a one-size-fits-all can't work really the same for all of us. So here is where that precision health comes into play, or should be coming into play in genetics. So in pediatrics, we deal mostly with prenatal situations, with the newborns and diseases that we diagnose in that pediatric age. And also we see a lot of adults with genetic disorders, many phenotypes present just later, that have susceptibility for developing a number of hereditary disorders, for example, related to cancer or neurological disorders. So today I'm going to focus on rare disorders. So these are, thousands and thousands of phenotypes that have been described around the world. We think that about 80% of them are genetic and about 50% happen in children. So you see that a lot of them would be coming and will be seen at the Stollery Hospital in pediatric age. And while they are individually, rare, cumulatively, they are common. And we think, and this is just a number from about 2013, I would say that number is going to just go up. But at the time, we thought that there are about 350 million people around the world affected by rare disorders. And that will be basically the population of the United States. So a patient that is diagnosed with a condition like that, or is born with these multiple congenital anomalies, it is a big surprise many times for the parents. And, the parents go through kind of a mourning process to try to accept this new reality of their life that they never thought of before. And because they are so complex, they are seen by many people in the hospital. Because if the child has a congenital heart defect, will be seen by a cardiologist. If he has a kidney disorder, will be seen by a nephrologist. If it is a hematological disorder, by a hematologist. And then there are all these people who come and have an idea about what is going on. In genetic, we are trying to see individuals as a whole and integrate all this information and evaluations from our colleagues, because the more differences we see in patients, the more likely it is that all these differences actually are related to a common cause that is likely genetic. So there are, like wrong diagnosis along the way, and all these patient odyssey that probably most of you have heard about, this roller coaster that the patient go through, explain why there is such a high emotional impact on the caregivers and the patient's lives. So in genetics, obviously, the DNA has been around for a long time. The concept of heredity has been brought forward by Hippocrates, the father of medicine, many, many years ago. And there have been many people who have struggled with this concept over the history of modern history of humanity. But I think that the event that has fundamentally changed the modern history and actually marked the beginning of this new specialty in medicine is 1953, when Watson and Crick have identified the structure of the molecule of DNA. Then in 56, the first karyotype was produced, and soon after the Down syndrome was identified, like that extra chromosome 21, these chromosomes that you can see under the microscope. A very big event in 1977, Sanger sequencing, that allowed us to actually read the text of a gene letter by letter, but at the time, not many genes were actually known. 2001, many of you will remember, we are celebrating actually the 20 years anniversary, I can't believe that, when the first draft of the human genome has been published with 20 years of work, hundreds of people working around the world, and $500 million spent on it. Since the 2000s, we have had access to microarray. And since about 2010, this new generation sequencing has invaded our world really like a hurricane. And we have been since trying to recover and trying to understand what this represents and how we can work with this technology. I remember very well in 2009, because I went to this very coveted conference, the workshop in malformation and morphogenesis. I went for the first time. I knew about it from the time I was in Europe, and I thought I would never make it. But here I was the first time in my life, and we had Michael Bamshad come, and he presented the gene for Miller syndrome. Miller syndrome is one of these classic genetic syndromes that are captured in this Bible that we used to use in genetics a lot, the Smith recognizable patterns of human malformation, but the gene was not known. And he presented the gene that was discovered through the use of whole exome sequencing. And I remember, the sighing, the gasping, we all understood that this is going to be a game changer, but I don't think anybody could have imagined what just followed the next decade. So just to give you a little bit of a sense, when I came here in 2012 in Edmonton, I started teaching, and I was just telling students how from 1953 to 2012, there have been 3674 conditions that have been characterized molecularly. And I just looked the other day, in the last 8-9 years, now we have 6,047 conditions. So basically in the last 8 years, we have an additional 2,400 conditions that have been characterized. And I don't know about you, but I'm not that smart to learn additional 2,400 conditions that are actually published, characterized, and almost every day, and to be able to recognize them in the clinic and make diagnosis. So obviously the world has changed completely, and it is, the progress has been far beyond exponential. So in genetics, like in any other discipline, we want answers, we want diagnosis, and we want to produce, to give disclosure to families. We would love to understand the natural history of disorders. For example, for Down syndrome, we have guidelines, growth curves. There is an entire clinic dedicated at the study for this condition. But for these other ones, where there are 5 patients in the world described, we don't really know what that natural history is. So there is a lot of effort needed to actually put all these people together and understand what these disorders mean. Ideally, we will stop investigations because we have found an answer. We will be able to provide management and anticipatory management, especially when the diagnosis happens very early, maybe prenatally or in the neonatal period. In some cases, we are able to offer surveillance protocols for patients. And in some cases, even drug therapy, because this has been the biggest challenge. Everybody says, oh yeah, but you are not treating in genetics. Well, things are changing slowly because obviously these are very complex circumstances. And then, from that N of 1, that patient, now that information can have an effect on the carrier status of the parents, but then there are siblings, there are aunts and uncles and cousins. And the things that trickle down, and there is this big impact in the entire family from our work. So, but how do we do this? We don't have a whole lot of tests. We have few, but very, very powerful and very complex. Just to start with the karyotype that I mentioned before, this is like looking at the 46 chromosomes, like at a library with 46 books. So when you look at the library with 46 books, how much can you tell about the books? You can count them, and maybe you can say that this book is a little bit wider or a little bit narrower, but not a whole lot. So in this way, we have for many, many decades been able to characterize about or diagnose about 5 to 10% of the genetic disorders. Now, in order to see something with a karyotype, you need a change of about 5 to 10 megabases or million letters to see that difference. We have now moved on to comparative genomic hybridization. We have replaced the karyotype with this new gold standard test, also known as microarray. And these tests, in an ideal way, it allows us to kind of pull each book out of the library and see if there are extra pages or pages missing. Because as you can have an extra chromosome, entire book extra or missing, so you can have pages. So for you to understand what the microarray does, remember when you are at the grocery shop and you have this magazine that you kind of just go through, and you don't have time to read the text, but you read the titles, you see the images, and that already gives you a lot of information about that magazine that you can decide if you buy it or not. So it is with this microarray. It gives us that kind of a little dosage information across the genome, and by replacing it with the karyotype with this technique, we are able to diagnose about 10 to 15% of the genetic disorders. So now we are moving from that 10 megabases to about 100,000 bases. So we are zooming in about 1,000 times. And then obviously, if we do the microarray and the microarray is negative, but there is a strong suspicion of a genetic disorder, we want to be able to sequence the DNA. So that means now that we can read the sequence, the text of the genes that are on like little paragraphs on the pages of the 46 books. And in this way, we actually thought in the few years ago that we will be able to kind of diagnose the difference, like 85% of the patients. We know that's not true now. And the yield of that test is somewhere about 0 and 100. It depends if you are catching the right gene, sequencing the right gene, or the technology allows you to read the right change in order to make that diagnosis. But we make that leap from 100,000 bases to the one letter that we can diagnose as a mutation. So next generation sequencing has allowed us to change completely the way we do testing, because in the past, we had this concept of single gene disorder. So you have one disease, one gene, and you go and you sequence that gene if that gene is known, and then if that is not known, you go to the next one. Well, next generation sequencing allowed us to be actually much more comprehensive in a more timely manner that also makes sense in terms of price. So we can go and test at once a group of genes, a panel, if we have a very clear diagnosis in mind. For example, short stature, there is a short stature panel. Or if the phenotype is not clear, there may be more than one diagnosis, which is possible, and we have too large of a differential, we can go and do a whole exome sequencing. So in one go, we can read all the 20,000 genes in the genome to see if we can find an answer. Of course, this represents about 1.5% of the genome, as I said before. So the difference will be covered if you are able to do a whole genome sequencing. Now, that is not available in the clinic. What we have available in the clinic today are panels and WES, but whole genomes is the direction the world is going towards. So just for who is interested to understand a little bit more about these pipelines, we collect that sample of DNA that is sheared in small fragments, and then we are trying to amplify every single coding segment of the DNA to amplify it, to make it actually readable. And that's through the sequencing, and then these fragments are mapped to the reference genome. There is an alignment of these amplicons, and there is every single letter is interpreted and annotated as in relation to that reference genome. And there are pathogenicity filters to interpret the significance of a change in relation to the reference genome if that is present there. So the results that we get from these tests are actually 5 classes: benign, likely benign. These are normal variation. These are not reported. The pathogenic and likely pathogenic, these are the ones we are after, because these results allow us to apply our results in the clinic, to use them for the management of the patient and their family. But very often, we do get these variants on unknown significance, where we are not sure what they really represent. And it's fantastically frustrating to see that you got a variant of unknown significance, and here is the phenotype of the patient, and you see the connection between the two, but you are not able to use that in the clinic because you do not have that ultimate evidence to demonstrate the pathogenicity. And just to illustrate with one thing, you need to really be sure if you offer a gene for a prenatal diagnosis, you really need to be sure of the significance of that test that you're offering. So here is where, we need that further evidence, which is very hard to obtain. It's not part of what we do in the clinic. This is where our work actually stops. So we want to be able to move that variant of unknown significance with some more evidence, either to make it pathogenic or to understand that is normal variation, and then we can move on and think about other causes. So, with the exome today, there has been a lot of hope, there has been a lot of progress, but we diagnose about 30% of the genetics disorders. So it's still a lot left out there, and I'm not going to go in the causes. We know that even if you take that negative exome that you have obtained from a patient and give it a little bit of time, and you do the reanalysis, an additional 10 to 15% of diagnosis can be found with these in this way, because genes are discovered all the time, the softwares for interpretation and the technology improves all the time. So this is how we actually are starting to think in moving forward. So applications of this technology, you can just go really wild with your imagination. Prenatal area, very dear to me. Yes, we have started a project just now in April using whole exome sequencing in ongoing pregnancies. This probably is going to be used at some point, in neonates, that instead of doing the newborn metabolic screen, we will do maybe an exome, and we will find all sorts of differences that will help us manage that individual. Of course, there are a lot of ethical implications. Again, I'm not going to go into details, but you can imagine that can come from these applications. And of course, pharmacogenomics is something that is a very immediate kind of application of precision health, which at this point in time has had already an impact in cancer genetics. So challenges that remain are multiple. And obviously, one is related to the population around the world. These kind of tests are usually expensive tests. Genetics is a discipline that has developed a lot, but mostly in the Western world. And the Western world does not represent the majority of the world. So we do not have information about what is normal in many people around the world. And even not to go too far from home, we do not have information about, for example, what is normal in First Nations. People who are here, with whom we know, are our friends and our many patients actually in Edmonton. Another challenge remains that interpretation of these findings. And this is where I think I want to raise your attention, because this is the direction I'm going towards and I'm going to talk about in the next few slides. And of course, in this digitalized world of medicine, it's very different the way we need to deal with storing information. This is not the paper charts that we used to carry around just a few years ago. So, these will be changes that will have to happen to the infrastructure of our medical system that have not been considered before. So why are these disorders so difficult to understand? Just few ideas here for you. Clinical heterogeneity. Even patients who have the same disorder in the same family look different from each other. There is genetic heterogeneity. Many disorders now have a multitude of genes that can lead to that disorder. So things are much more complicated. Incomplete penetrance is a very hard concept to understand, but there are people who carry these mutations, but they do not show features. So that will make it very difficult for us to interpret even a pedigree, for example. And mosaicism is another phenomenon where the mutation happens in a part of the body. When we do genetic testing, we do it from a peripheral sample most of the time. And these are the DNA from the white blood cells. Now, if that mutation is not affecting the white blood cells, your test is going to be negative. So these are all things and thoughts that we need to have in the clinic when we assess patients. And what we are looking for that biological evidence, starting with the genomic structure. What is the expression pattern of what we are witnessing in the patients? Because genes have tissue-specific, time-specific patterns. What are the physical interaction and the functional interactions between these molecules? No protein is hanging there alone, but actually acting in a concerted way with other proteins in pathways. And these make, very in a simplistic way, the complexity of the biological processes. So how we put all this information together relies on how good our interpretation is, what we see in the clinic, through investigations, through blood work, through imaging, and how good the evidence is from the biology. And many times these are not coming together, and many of the evidence is missing. So the American College of Medical Genetics has come up with some framework for interpretation of these variants to help our colleagues in the labs when they call these variants. And this is not a table to go through. I put it here just to understand how much evidence our colleagues in the labs are looking for when they call these variants for us. They look at population data. They have a number of in silico programs to assess pathogenicity. Whenever available, functional data is very important. We do segregation studies in the clinic. It's important to know if the mutation is de novo or is inherited. We look at allelic data. We look at a number of databases that have been developed in normal and abnormal individuals to help with this interpretation. However, many times that biological evidence is not there. And this is where we stop, as I said before, our work in the clinic. So there is a push to move into that realm of research and try to provide that functional evidence, that biological evidence to improve our diagnosis. And the same college has come with some ideas around how to deal with this. And you can see that top of the line here. And the fundamental thing that we need, and we cannot go around, is to understand the disease mechanism. As far as we do not understand the disease mechanism, we cannot dream of moving forward to therapies. So how do we do that? We need to use assays that are around. They are used and practiced every day by our colleagues in research laboratories. We just need to make sure that these are validated, and then we can use them for patients for the interpretation of these variants. So in Canada, there have been enormous efforts to discover genes, to understand disorders, and the trailblazer has been Kim Boycott, our colleague who trained in Calgary and now is at CHIO, who has created these enormous opportunities across the country for all of us to access these sophisticated tests and try to find the causes of rare disorders. Now, these projects have had a number of iterations, and at every time, Edmonton was part of these. Before me, these were led here in Edmonton by other senior colleagues like Dr. Orden Lehman, by Dr. Oksana Sukorski. Nowadays, we have Care4Rare. We are up and running here in Edmonton. And I'm particularly happy to see that we have now the silent genomes up and running. It's a project initiated by our colleague, Dr. Arbor in Victoria, that is looking to accumulate information about the First Nations. And this is a very big step forward because the First Nations leaders have been able to be engaged in this project and work together. Another project led locally by Dr. Kanu is this methylation profiling, is a genomic test to look at patterns of methylation that are specific to different genetic disorders. A project that is initiated from London, Ontario by Dr. Sedikovic. And the Tiger, this is a provincial study that is initiated by Calgary, but it involves also Edmonton, that is planning to repatriate the exomes, which we now do, we now outsource, and then evolve over time to the use of an application of the whole genome sequencing in the clinic. But the question is, there is so much work, there are so many patients, there are so many questions. What does Edmonton do? What does Edmonton, makes its own and develops to take care of the local patients? And this is a thought that, I had for quite a while. Just a very, very brief, but just to mention that treatments are starting to enter the realm of genetics more and more in the inborn errors of metabolism, but even in other Mendelian disorders. And, this central dogma of the mRNA, the transfer to proteins, is actually applied to therapy. So all of you know that nowadays the mRNA therapy is important because we have vaccines developed to help us go through the pandemic. But they're also applied to genetic disorders, like the example here in this picture of the spinal muscular atrophy, a new therapy that is used for these quite common, actually, genetic disorder. And on the right-hand side, you have this document from the Parliament of Canada. It is important that there are these steps starting to evolve in addressing the therapies for rare disorder and access to treatment, because many of you will know how difficult it is to get access to treatment for patients with rare disorders. And with this, I'm going to move forward to the project, one project that I did here at the University of Alberta. So this project started very soon after I joined here in 2012. Is a patient that I seen in NICU who had a previous sister who died with a very severe muscular condition, and the parents were consanguineous. So there was clear to me that this is a genetic condition, but at the time I didn't have even the clinical tests to be able to advance very much. So I needed a little grant to be able to run a SNP array and look at homozygosity regions. And then I got an innovation grant. So through that, I got access to consents for engaging patients in these kind of projects. And then in the summer of 2014, got a scholarship, a student, and we found a candidate gene. We did a segregation study. So we were very excited to find all of that, but then everything stopped. And I went for a coffee with a friend of mine, and I was telling her about this project, and she says, "Oh, I have the people for you." So that's how I got in touch with Emmanuel Cordat and Elaine Leslie in physiology. We got, soon after that, the seed grant, and we had a wonderful collaboration. Few years of works with students, to complete the functional studies and find that evidence behind our disorder. Obviously, what happened usually is that others will be faster, and the gene was published, and other people published other patients, other functional data. But eventually in June of 2019, we published our paper, and obviously we were very happy about it. But there was something that was bothersome, because for a project like this, 6.5 years, it is really unrealistic. By the time we finished this, our family was beyond the reproductive age. So really did not have an impact on them anymore. It may be used by other people around the globe, and is very good. It's a very good outcome. But we really want to have that impact on our patients that we see here at the Stollery. So, this has open opportunities for my collaborators to study other mutations, and maybe they will become the center for, focusing on this disorder that we described. But I think we can actually do more. And thinking about the way this went, it went actually pretty well, because, we got money, we got students, we got fantastic collaborators. So we actually had all the ingredients coming coming along. It's just that that time was extremely, extremely long. So I've been thinking about these, and I would say that if this happened today, it will be different because the technology and the access that I have in the clinic is different to do testing. But there are some elements that are kind of recurrent in these projects. Like, for example, ethics consents for studies. Funds, yes, these, we had funds, but this remains a big challenge for anybody who wants to engage on a project like this. Collaborators, I've been extremely fortunate. All my projects that I'm running here at the University of Alberta are done with local collaborators. So I got to learn in like first experience, first person, that there is a gold mine of of expertise that we have locally, and students. Extremely important to find students that are interested to study these disorders and mechanism of disease. And this actually is, so it takes a lot of effort to bring this boat or this ship into port, and then many, many pieces to coordinate in order to be successful. And this is what actually brought me to, talking to Todd many times and talking to other colleagues. And I know that, the way things are happening are discouraging many people who probably have very good findings in their patients, but everything is so difficult to do that it is just easier to just give up. We are all busy. We can't spend this time with every single variant that we find to try to find evidence. So this is the idea of this hub. This hub is meant to be that bridge builder between the clinics, the stollery, about all these findings that we find in patients, connecting them with trainees, and connecting with research, and facilitating that collaboration between these areas to make these projects successful. So I have to thank Tanya, is our coordinator, who helps us, imagine all these things. And here is a framework for the hub at this time. We hope to be able to have these interesting findings or strong phenotypes that suggest genetic disorder coming from the clinics here at the Stollery, that will be first reanalyzed. As I said, this is the first step that we take nowadays when we have negative exomes. And if interesting findings are coming through, or a candidate gene is identified, then that will allow us to do that intake into the hub and try to provide all these resources to help push that project forward. So in more detail, this first step, we'll have a negative exomes from any of the clinics, or specifically from the clinics that do exomes, like the medical genetics clinic, and a strong suspicion of a genetic disorder. We will provide the referral for the hub. We have a research coordinator that can consent for this first step and will engage in reanalysis. Reanalysis sounds easy, but is actually quite a complex process that requires access to software, bioinformatics support that we do not really have locally, but it can be even outsourced. In today's world, things work different. Access to databases that are essential. And we are working right now in developing that local multidisciplinary team to be able to do this reanalysis. This technology is here to stay and just become more complex as we move towards genomes. And I think there is a role for us here in Edmonton to develop that expertise. Now, obviously, in this new system, we need that storage capacity that looks very different from what it has been in the past. And I'm very happy to say, and thanks to Dr. Richer, who's offered to help us with that. Now, what comes out of the reanalysis is if a positive diagnosis comes out, that will be fed back to the pediatrician. And if a negative result is found, but there is a variant of unknown significance that makes sense in the context of the patient, or we find a candidate gene, that opens the possibility of intake into the step 2. And step 2 would be these information that will come, from directly from the clinic, from the medical genetics clinic where we do exons, or from the reanalyses where we have these strongly suggestive variants that would have some potential for management implications, or a candidate gene that can be referred through the research coordinator to the steering committee of the hub, where we will encourage clinicians to submit to GeneMatcher, another fantastic resource that we have nowadays, which is an international database where you submit your gene of interest and you can connect with other people around the world who have patients with mutations in that same gene. We will help clinicians to develop that, put together that information that provides evidence for their case, and will help and invite that clinician to present their case to the advisory committee and make that link. The advisory committee would look at this evidence, and if they think this is a project that has legs, that could be recommended for intake into the hub. We will have a consent for this second part of the whole process, and we will be able through the hub to provide sample storage, to make that connection with a basic researcher. Hopefully that will be a local colleague. If not, we can try outside connections. We'll provide a means for funding opportunities and a student. And through that process, that information should go back in the end to the pediatrician. So we will encourage, if a strong candidate gene or project is identified, to access other resources that are available. For example, these models and mechanisms grant through CIHR that provides $25,000 to develop an animal model. Very important when we are looking for evidence for disorders. Now, ethics, starting from the beginning, ethics, fundamental, consents, we cannot move, and we can't even envision a project without that. But with the help of Dori and Farah, we are working through that process to obtain these umbrella consent, so that not that every clinician will have to go through an application, but will have that umbrella consent to allow them to enroll their patient. Biobank, extremely important to be able to collect samples from patients. And we have an extraordinary resource here in Edmonton through the Biobank of Dr. Bruce Ritchie. Sample collection obviously is always based on availability, but also depends on the gene expression pattern of that specific gene that we are trying to address. But Dr. Ritchie also can offer immortalization of cell lines and storage of the samples. The advisory committee. The advisory committee is really central to the success of this hub, because really we are looking for people with expertise from across the board, because every system and every organ in the body can be affected by a genetic disorder. So anybody who thinks that could contribute to this is welcome to contact us. We would like to develop a list of individuals. We have talked to some colleagues who have expressed interest already. And really, people with expertise in any area of biology could become essential based on that specific project that we are looking at. And integrating data, I think it's a dream of many of us to be able to connect behind a secure firewall, our databases from research with information from the electronic medical records, integrating features of the patient's phenotypes through these new program phenotypes that is actually now worked on, to have that storage data available, and to make this whole process that is so complex really smooth. And just like a final slide to put all of that together is a little bit of a busy slide, but I'm just trying to put together, all these pieces, moving pieces in the hub that we are thinking of and we're trying to connect. Primarily the clinicians with the researchers, and just thinking, wouldn't be nice to have this, an app where you can enter a name of a gene or a pathway or an assay that you're looking for, and then you will have a list of people here who are doing that assays, that are working on these pathways already, so you can quickly access them and get in touch. Put together trainees, have these consents, have access to biobank, have access to bioinformatics, have access to a number of funding opportunities to make these become a reality. So I think that I quickly summarize, the enormous progress that has happened in our field, especially in the last decade. I try to present research and the challenges of what a genetic research projects can be, and introduce to you this idea of the hub. Before I finish, I would like really to thank Tanya, Dori, Farah, and Todd. This obviously wouldn't happen without you. Peter has been an advisor for us. We are very grateful to all the foundations that are helping, and to our friends at UBC, Dr. Anna Lehman and Stuart Turvey, who already have in place a similar initiative and who have been an inspiration for us. So here is, another idea for the hub, this boat with many oars. So we need many people to take one oar each and contribute together to the success of this initiative. And before I really finish, I just wanted to close with this familiar image to most of you here at the Stollery, this molecule of the DNA that is centered in our hospital. It was here when I came to interview in 2012, and I took it as a welcome, and it's still here today. So it is my belief that genetics is to biological sciences what anthropology is to social sciences, because the DNA is that unifier element for our shared human nature, but not only, also I hope for our shared experiences and hopefully for our future projects in the hub. Thank you.
Thank you, Juana. Interesting and still lots to do, I see, and we're getting there though. I'm excited, and I think one of the things that we should mention is that if people are interested in getting involved, please reach out to either of us. The more the merrier, as Juana said. I guess if you have questions, either click or go to the box. I'd like to start with a quick question. So I've noticed the nomenclature and the words around. We use variation, and we use variation of unknown significance. We use variation that's likely pathogenic or pathogenic. We stopped talking about mutations. Can you comment on the use of the word mutation? Is it no longer in vogue? Is it offensive? Or how do we refer to these?
Well, a mutation is something that is really pathogenic. We get a lot of variants in the DNA. We have a lot of variants and they have a lot of significance. So, a variant that is pathogenic, I would equate with mutation, but it was decided that you should use these word variants. It's not my decision for all these changes that we see. As I showed, there are a variety of variants that, and many of which I did not talk about.
Manuel, I see you're unclicked. Do you have a question?
Yeah, I was typing, but I think it would be faster to just ask the question. I really love the idea. I think it's a great idea. And I think it's really that should help so much. And so what I was wondering is, Juana, how much shorter do you think conducting a project from bedside to bench back to bedside will be compared to what we have experienced together, like the 7 years from the discovery of the mutation to publication and probably back to the patient. So how much shorter do you think this will be using the hub that you are building?
So, I think it's very hard to be, give you a number, because, every single variant or every single candidate gene, as you know, requires work. Some will require more, some will require less. Depends how much we want to get involved and how much we want to support through the hub. But really, many of the steps that we have gone through, I think, can be shrunken. And this is what I'm trying, with these little pieces that I presented, that we are trying to cut a little bit of some of the corners. The only corners we cannot cut is really the work that has to be done with the biological evidence that we need to build, where we need to be very rigorous. So that is something that you are a researcher, how, when you start, you never know when you end.
Yeah.
But, I'm hoping that at least some of these steps can be shrunken. And then, if there is more expertise and we are able to make these connections with more facility, all these can actually contribute. And end of the day, they add up and hopefully will make it more real, because it has to be real for the patients. When you spend 7 years on a project like that, as you see, it's not real for the patients anymore. So we have to really think very hard, how do we want to really participate in that process? And this is, a question, that goes so much beyond me, because I am not a decision factor here. I'm just, bringing my little experience over the last 8.5 years in Edmonton. But really, I am really open to anybody who has heard today this presentation, who has better ideas, who can make suggestions, critiques. I'm really open to everybody. I really want this to be a forum where we meet and work together, clinicians and researchers.
I think it can be cut in half, Emmanuel, because you'll eliminate writing de novo ethics. You eliminate this sort of random factor where somebody has to know somebody and you have a coffee to run into the right researcher, right? Because hopefully you have people that can make those connections.
Yeah.
And then you have all the resources in place to move the patient resources to the researcher. So I think I'm thinking about half at least.
That would be great. I mean, the other thing I was wondering, again, based on our experience, like we really love that we had the support from WCHRI, where what was difficult on our side was identifying a student and finding a student who would be a graduate student who would be in the lab for long enough to perform the work that would be needed. And I see in your slide that you have a pool of students, and I would love to have a pool of students, where we could connect and ask them, are you interested in working on this project? But the reality is that we are really struggling to find good graduate students who are willing to work in a lab. And I don't know if WCHRI could help there, but if it's a matter of advertising better or more, going to advertise to different pools of students, but in the Faculty of Medicine, this is really difficult.
So I see that Ing Sui has raised her hand. So
Hi. Yeah.
She may help us with something here. I don't know.
What's that?
Maybe you're helping us.
Oh no, I thought that was such an exciting talk. I was really, really carried along, and it just opens up so many opportunities for the patient and opportunities at the other end of your link of chain. Researchers, brand new projects, and I think the idea of reanalyzing data as new analysis pipelines are coming through is just really what one should do with that amount of data that's there and not waste it, but you know what I mean, like an unknown variant. So, and anyway, I'm just kind of rambling, but I would love to help and be part of it. Like, I guess I'm saying it in a public forum. I can't back out now, but yeah. I'd love to help just kind of connect. Yeah, really. It's just us talking, right? So I think that
you're going to mention MD STIR, because that's a group of students that potentially could be interested to participate in these projects.
Exactly. So definitely the undergraduate medical students that are very interested in research, looking for exciting projects for them is really great. And I think Manuel's comment is really important too, that graduate students will really be able to carry sort of the really large, larger project or larger load. So I kind of did actually have a question a little bit. Like, I really like the concept as well of all these different projects that can come out of it. And my question, of course, is like, not of course, but like the role of race, right? So the reference genome and now even like in the breast cancer world where I'm looking at, the role of race for even BRCA signatures and such. that are defined by the genomics. So I think that's an exciting area as well, right?
Yeah, so this is obviously we conceive these in the realm of pediatrics because I mean, my connection is natural to Stollery, and I do spend my time with pediatric patients, and we are supported by the foundation. But if this is something that makes sense for people, this can be translated very much into the adult world, and we need to make that connection because actually, you know what? A majority of the referrals to medical genetics today are actually from the adult world, and you will know better than me how much progress and how much impact next generation sequencing has had in cancer and modified therapies and so on.
Absolutely, yeah. So exciting, thank you.
Yeah, thank you.
There was a question here from Georgina McIntyre around training opportunities for students. So there seems like there's interested students in the analysis piece. So currently in the analysis piece, sort of whole exomes and whatnot, we are in a building stage. So we're looking for opportunities to recruit and expand there, but there will be, and we are creating, as you can tell from Dr. Cordat's and Goping's questions, opportunities for students to get involved. Yes, there will be lots of opportunities for students, maybe not immediately in the data analysis piece because that's not really our strength currently, but it's an area we're building in. With that, so there's a question from Charlene Rutherford about the input of women's and women's health clinics into the hub. So yes, absolutely. I think, Charlene, there will be. We're starting probably with very young children because it's sort of more. You got to start somewhere, but obviously, as Dr. Goping mentioned, there will be specific women's health diseases that would link well into this and patient populations at some, probably not so future. I think we'll just start with children's health and move to that. But I mean, if you think about the antenatal piece, which is a very clear piece, that will be involved right away because a lot of the input will be from the antenatal health.
Yeah, so we are developing our prenatal genetics clinic at the Royal Alexandra Hospital, where we do prenatal genetics, and really we, see women and in pregnancy. So we see women, we see their children. For us in genetics, we do not see this separation because the children and the women are related through their DNA. So for us, really seeing them, we see them together as a unit. And I'm just hoping that, usually these projects do have an impact for the women because the women wants to know, what is my chance to have that happen? Am I a carrier? What is our chance? So, this connection is there naturally.
Yeah, it's tough to explain genetics. You have your DNA your whole life. And even, yeah, so it's a cyclical thing. With that, if there's no more questions, we're well past the hour. I thank you again, Juana, for this talk. It's been a pleasure working with you, and it seems it was well received, and a lot of people are excited by it. So thank you all for attending. Have a good afternoon, and we welcome you to the hub.
Bye.
Take care, everyone.
Thank you.